Hi, and welcome to Nothing is Rocket Science, a podcast where we talk to different professionals, scientists, educators, and science communicators to understand the nuances of science behind everyday objects and concepts. Today we have with us Miss Kerry Silva McPherson.
Mhm.
She holds a BA in biotechnology from Assumption College in Massachusetts and an MS in cell and molecular biology from the University of New Haven in Connecticut. Um, Her master's research explored the relationship between breast cancer progression and mesenchymal cells of the tumor microenvironment. Currently, Kerry attends Yukon Health, where she researches proteins involved in chemotherapy resistance, and she's also a PhD candidate and is interested in a career as an MSL or as a science communicator. She began the Bolded Science blog, which has probably over 50 posts now in the last 1 year, I guess, 1.5 years. Um, it's a collaborative blog that gives scientists of all career stages a voice and a chance to practice writing for a non-scientific audience. And that's where I actually met Kerry, although virtually. I also contributed to her blog, and we've been working with each other maybe like sporadically, but yeah, we've been in touch with that because of that. Um, so on Bolded Science, grad students, research technicians, or undergrads can write about their research, a scientific topic of interest to them, or even about life as a scientist to get published on the site. So we're going to talk more about this and more about cancer and cancer research with Kerry. So welcome to the podcast, Kerry.
Hi, thanks for having me. This is my first
Yeah.
podcast experience, so it's exciting.
Oh wow.
Yeah.
I'm glad it's mine. Yeah, before we head on to these heavy topics, uh, what made you pick cancer research? Like what got you interested in something really morbid?
Yeah. Well, at the biology level, I don't know if I would describe it as morbid. Like, obviously, it's a very serious disease that affects a lot of
Hmm.
people, but on a daily basis, that's not really your thought process of thinking of it. But I think a lot of scientists, and I'm not sure if you were like this a little bit, your research kind of almost chooses you rather than you choosing your research. So
Hmm.
I actually always thought that I would study Alzheimer's when I was an undergrad. I always thought that that was going to be my disease of choice. I thought it was really interesting. And I think when I got my master's degree at the University of New Haven, I was actually hired as a research assistant in a cancer biology
Right.
lab. So for that lab, I didn't choose the lab. My PI chose me because she interviewed me and wanted to hire me as a research assistant. And
Right.
I don't know, I just like science ever since. I think it's a really complicated disease that has a lot of factors to it.
Right.
And, you can also study it in different contexts, whether it's you're studying breast cancer or brain cancer or pancreatic cancer. But then I've also was fortunate to study at it as a cell biologist and now as a protein biophysicist. So overall, like I actually have seen and been involved in research with cancer at multiple levels. And I think at all levels, it's actually a pretty interesting disease to study.
Hmm.
Mhm.
And so when are you right now, you know, do you is your research narrowed down to any kind of cancer, or are you still, pursuing breast cancer? And is it still more about breast cancer, like your master's thesis?
No, so actually now, oh, are you still there?
Yeah, yeah, I am.
Okay,
Yeah,
sorry,
sometimes
the
it's
thing
just
went
yeah,
away.
the video. Hmm.
Yeah, so as of now, I am actually just looking at cancer in general. And we'll get into this in
Hmm.
more discussions later, but pretty much I study proteins that, when overexpressed or hyperactive in cells, they can cause
Hmm.
cancer to become resistant to very common chemotherapies.
Okay.
So one of the most common chemotherapies that's used in treatment is a chemotherapy called cisplatin, which has been approved since the 1970s and it's been used ever since. And it attacks cancer cells pretty much just by damaging their DNA so that they can't replicate, so they can't keep growing.
Mm-hmm.
And you can use this drug to treat all different sorts of cancer. So it's not used for just breast cancer or just prostate cancer.
Right.
It's used for all sorts. It's actually used in 50% of chemotherapy treatments.
Okay.
So, it's a pretty blanketed chemotherapy that can affect
Mm-hmm.
any rapidly growing cell. So that's pretty cool because I'm not zeroed in on just breast cancer or, a certain kind. I'm researching something that could affect all cancer treatments.
Right. Yeah. So
Mhm.
it's not it is kind of a blanket treatment and not just aimed towards one particular kind
Mhm.
of cancer. Right. Right. Um, yeah, if you can briefly tell us what is cancer and what is happening at a cellular level in cancer.
Right. So, at its most basic definition, cancer is just unregulated cell growth, and that can
Okay.
cause, and if it's a tumor, for example, that would be a solid tumor growth, but also there are some blood cancers where you just have uncontrolled growth of cells as well. So, pretty much this uncontrolled growth is going to occur when your DNA is damaged to the point that it causes your cell cycle or the mechanisms of a cell that regulate the cell cycle to go haywire. So our
Mm,
cells need to replicate to grow or to replenish certain organs in your body. Our GI tract is constantly being replaced with fresh cells. We get new red blood cells and white blood cells because of cellular replication. It's something our cells do. But there are certain cells in our body that shouldn't replicate or shouldn't replicate as it should be controlled, in other words. So they can't just replicate forever on end. They replicate when they are meant to. So sometimes our DNA can get damaged where it causes proteins to make the replication go too fast. And that can
mm,
cause tumor growth. Another way tumor growth can also happen is if we have certain mechanisms in our cells that cause our cells to die. So cells don't live forever, but sometimes our cells can also go haywire where they don't die. So then you get a buildup of cells. So
mm,
those are the two most common ways that this uncontrolled cell growth happens. And in the end, it all comes down to DNA. So if your DNA is damaged, so it doesn't code for your proteins in your cellular processes properly, that can cause this uncontrollable cell growth. And that DNA damage can be something you inherited from your parents. It can be a chemical that you were exposed to, such as like asbestos or cigarette smoke or UV light. Or it can even just be, our DNA just gets damaged all the time. And, when we eat food, we have metabolic processes that happen in our body that can damage our DNA. And sometimes if it
mm.
damages the right gene or if we aren't able to fix that DNA damage, cancer can just happen. So sometimes it's not always rooted to a specific event or of
Yeah.
genetics or environment. So, unfortunately, if the right genes get mutated, it can cause cancer.
Mm. And so, how is a malignancy different from benign? Like I understand that malignancy is a little more severe, and benign is it's there. It's like a silent thing there that's in your body,
Mhm.
but it's not causing major problems other than the
Right.
fact that there's something happening in your body. But malignancy is a little more serious. But again, at a cellular level, how are they different, and what are the repercussions?
Right. So, as I said, the definition of cancer is uncontrollable cell growth, but that's not its only characteristic. So, cancer cells can also mutate in other ways to do some pretty scary things in our body. So,
Mm-hmm.
one of the ones that's scary is that is metastasis, which is just a
Yeah.
fancy word for spreading. And I think
Mm-hmm.
that for many of us who have had family members or you have personally had cancer, that's probably something you might have heard of. Like if you have breast
Right.
cancer, but it spread to the lymph nodes or the lungs, that's when you understand that it's very serious. Or even a malignant cancer may not have spread yet, but it has the potential to spread. So that is
Mm-hmm.
definitely one of the things that could be a little bit scary. But they can also do all these other things. So cancer cells, because they're growing so much, they want to take up a lot of the nutrients from our bodies in order to
Mm-hmm.
continue growing, correct? So that's another sign of malignancy where it is altering its metabolism, but also the metabolism of our healthy cells. So it can just use up all the nutrients and the oxygen in the region. And that's another form of malignancy. Another thing a cancer can do, especially solid tumors, is that they can actually signal for blood vessels to be reworked towards the tumor. They call this angiogenesis.
Right.
That's a pretty scary thing to go on to, because one of the treatments of cancer is surgery, and that can complicate the surgery. Further down the line, another thing, and part of my master's thesis touched on this, and this isn't as well of a characterized trait of cancer. The cancer cells can do something called cachexia, where it actually sends out signals to kill your muscle cells, and it can cause you to become weak. And the reason it does that, again, is that way it can absorb nutrients in the area rather than our muscle cells being able to take those nutrients. And then also the positioning of the tumor also matters, because it can interrupt and press against our vital organs.
Right. So this all happens in malignancy, right? So in the stages of malignancy.
Yeah.
Great. Predominantly, all of us, I think, we've come to understand that hereditary is like, genetics and hereditary is one of the main reasons to be at least cautious of. Like, you have a family member or a first immediate family member who's had cancer, so you know that you're genetically predisposed, and you can either be careful or at least go get yourself tested, and you're at least cautious about it. But now we also have new types of cancer, or like we've had that in our family where maybe my grandparents, the generation of my grandparents never had any kind of cancer, but I've had people like family members who've developed cancer. So what is the role of environment in causing this mutation? Like, what are the different causes? So we know hereditary is one, and what are the other causes you think that causes cancer?
So any type of exogenous or endogenous chemical that can damage our DNA is something that could cause cancer. So let's take UV light, for example, from the sun. So the reason why UV light can predispose you to cancer, specifically skin cancer, is that it causes some of our DNA bases to covalently bind to one another, which isn't something that's really supposed to happen. So then we have certain proteins in our body that can repair that damage. But then if those proteins don't repair the damage properly, then you can have a mutation, which may cause cancer. So interestingly, when we, you can't almost look at environment and genetics apart. So as I said, we have all these proteins that can fix DNA damage. But if you are genetically predisposed to have a protein that doesn't fix the DNA damage very well, you might be more likely for that environmental effect to cause cancer in you.
Right.
So it's really hard to just tease them out separately. But in the end, when it comes down to environmental mutations, it all comes down to something that's either binding to your DNA or maybe binding to a protein and making a protein go haywire, not doing its job well when it comes to a cell cycle or DNA replication. And other examples of that are obviously cigarette smoke, chemicals that you might be ingesting, whether that be in processed foods or maybe even if you don't have clean water. Also, viruses are a very common something that can infect you, that can predispose you to cancer, because viruses hijack our cells and hijack our cellular machinery to replicate its DNA. And in doing that, sometimes it can mess with our genome and certain cells. So, and HPV is the most, I think, famous example of that, of a virus that we know that can predispose you to cancer. So that's another example.
Oh, wow. I mean, speaking of viruses, do you think COVID, is there research going on where COVID also alters this kind of thing, and or is it kind of a risk factor for causing cancer?
So I don't know if that's known yet. So it's going to be hard because usually cancer will not be diagnosed until much later down the line.
Right. Right.
So yeah, that would be something that we're going to have to wait to find out, but I hope not.
Hmm.
But it
Yeah,
is
hope not.
possible. Yeah.
Right. Yeah. I mean,
Yeah.
with the number of variants that we're getting with time, I think, yeah, like,
Yeah.
basically the protein's getting damaged, like you just mentioned.
Yeah.
Hmm, right.
Because that's another
Um,
important. Oh, sorry, but
yeah, yeah,
that's another
yeah.
important point that I kind of haven't brought up yet either, is that it's not just one genetic mutation that's going to cause cancer in your cell.
Hmm.
It's often about 2 to 3 different proteins. I mean, excuse me, genes in combination with each other if they're mutated that will cause that uncontrolled growth. And then as your cells begin to grow, they are already growing in an unstable genome and really fast and in under stressful conditions because it's using up so much energy. And then those cancer cells can accumulate more mutations as the cancer cells continue to proliferate and grow.
Hmm.
So that's probably, I think, one of the biggest misunderstandings, especially when we talk about the genetic factors. Some people will say, well, my mom had cancer, so that means I'm definitely going to get cancer. Not necessarily true. So you might have that one gene that predisposes you to cancer, but it's going to take 2 or 3 more events in those cells in order to form cancer to actually happen.
Okay.
Mhm.
Yeah, that's really interesting because, I mean, although we see that, uh, the percentage of people who've had a family history, like, if like you said, like if a mother has it, then there are very high chances the daughter might have it. But there's still hope that the daughter might not have it, right? So
Mhm.
I think that's great to know. Like, I didn't know to what extent. And is there a way uh, to not get it? Like, if
Mhm.
can we avoid it? What ways? Like, I understand the healthy lifestyle and stuff, but then there's something beyond that too, right?
Yeah, I think when it comes down to it, it's just going to be prevention, especially if you know that you're genetically disposed and just catching it early.
Mm,
Unfortunately, at this point, there's no like foolproof way to avoid getting cancer.
right.
But yeah, just especially if you're genetically predisposed, like your doctor will tell you to get more checks more
Yes.
often because the sooner you grab it when it's just in like a benign stage and it's not spreading, or if it's not really affecting your body too much, the sooner you can take care of it.
Mm, right. Yeah, like prevention and early diagnosis is definitely the key for, I think, any kind of disease. Uh,
Yes.
right. Um, Coming to, the cancer treatments, traditionally all of us are aware of like chemotherapy and radiation. So if you can tell us a bit about these two treatments, and I also want to understand, the treatments. I know, like, with time and with technology, we do have, a lot of new treatments available. And if these treatments still fall under these two traditional categories, or do we have any, new categories to treat cancer? Yeah.
Okay. So you're right. The two biggest forms of treatment are chemotherapy and radiation, but also in combination with surgery. So surgery is also key
Right.
for removing the mass as well.
Right.
So the way radiation and chemotherapy works is the hardest thing about treating cancer is you have to kill the cancer cells. That's the goal, is to get rid of
Mhm.
the cancer cells. But the
Right.
hard thing is that a cancer cell is very similar to our cells. So
Mm.
maybe they have 3 or 4 mutations that cause them to act differently, but with the exception of those 3 to 4 genetic mutations, they're identical to our own body and they have our own genome and our own proteins. And it's really hard for us to specifically design a drug that only kills the cancer cells, but on our healthy cells. So when
Right.
we think about, say, we have a bacterial infection, it's really easy to kill the bacteria because the bacteria cell membrane and genes and proteins are different than us. So we can just take an antibiotic, it kills the bacteria in our body, but we go on living our lives because our mammalian cells are not going to be affected. So there are a few ways that scientists over the years have designed selectively killing cancer agents. So
Mhm.
the biggest difference, the most common difference between cancer cells and healthy cells is that cancer cells divide quickly.
Right.
So both radiation and genotoxic chemotherapy work in a similar way, where we damage DNA. And when you damage the DNA in these really fast proliferating cells, the proteins cannot fix the DNA fast enough to continue to replicate. Because every time a cell divides, it has to replicate the genome. So if you damage the genome, so the genome can't be replicated, the cell is going to die. So that is the most common type of chemotherapy, and that's how cisplatin works. That's how radiation works. That's how a lot of alkylating chemotherapies work. And they're either agents that directly bind to the DNA to cause cell replication to crash out, or for radiation, it's actually just targeted radiation that breaks apart the DNA in half. So that way the cells can't replicate further. So that's the most common type of chemotherapy. It's just attack the DNA so the cells can't replicate further. Now, this is only somewhat specific. So often people get sick when they undergo chemotherapy. And that's because we have other cells in our body that do replicate fast, similar to cancer cells. So I think the most common symptom that we recognize in cancer patients is they lose their hair and their nails thin. That's because our hair is caused from cells that rapidly divide. So unfortunately, we're killing cells in our body that rapidly divide. We're going to kill those cells that support our hair growth. So that is the side effect of this kind of chemotherapy. Another side effect is a loss of appetite and a loss of energy. That's caused because our GI tract has rapidly dividing cells that help us metabolize our food. So those are going to be hurt by that kind of chemotherapy. And then another third very common symptom is a depleted immune system.
Immunity.
Right. And that's because our white blood cells are generated from rapidly dividing cells in our bone marrow. And unfortunately, those are getting hurt from the chemotherapy too. So that's the most common version of treatment, is just attacking the cell cycle and making all fastly replicating cells die off. So a lot of research right now is going into trying to make more specific chemotherapies that don't have as toxic of side effect and are more specific to the specific cancer that you have. So a lot of cancer right now has to do with understanding the specific genes that are disrupted in a lot of cancers. So one of the most common advancements that's happened recently is with breast cancer. There's a gene called BRCA deficiency. And BRCA is a protein that helps us fix our DNA when it's damaged. So a lot of breast cancer patients have a protein that is deficient so that your DNA does not get fixed properly, which can cause mutations, which can cause cancer. So scientists have developed a set of inhibitors that will only affect cells in a patient cancer's body if it has this BRCA mutation. So then therefore, this chemotherapy, which are called PARP inhibitors, and there's a whole family of them that were released a few years ago, this inhibitor is only going to affect the breast cancer cells because those are the cells in the body that have this BRCA mutation. So that's the general pathway that a lot of cancer chemotherapy research is going towards, like finding out genetically what is wrong with this cancer cell, and then using that information about the way this cancer cell was able to form and what it is surviving on, how it's able to propagate specifically according to its genome, and then designing inhibitors for that specific mutation that causes cancer.
Interesting. That's
right.
Yeah, that's a good piece of information. So are these so when you say chemotherapy, is it is it medication, like is it a tablet, or is it something that's given through an IV, or is it some kind of a radiation? Like, so what is chemotherapy? Like, I've read, I don't know a lot about cancer and its treatments, but I've seen patients, like even in my lab, we deal with some patients who have cancer who are actually undergoing chemotherapy on a daily basis. So I've come to know that, some of them are on medications, like oral medications, and some of them are on radiation and, blood transfusion, that kind of a thing. So how is it different? Like, what exactly is chemotherapy and radiation?
Right. So chemotherapy and radiation are often given to you as like a cocktail. So often
Mm,
when we receive treatment for cancer, we don't just get one drug. You actually usually get
okay.
like two or three. So
Mm,
like radiation isn't given to you and like is actually done to you. So it's not like a physical drug. It's actually
right.
like an agent that's like targeted at your specific cells. So that's a little bit different than chemotherapy. And then you're right, chemotherapy can come in different forms, whether it be an IV or orally, and it could be daily or weekly or what have you. But it's going to be specific to every patient
Mm,
because everybody's cancer is different. Everybody has
right.
different mutations, and their doctors will predict which cocktail treatments will be best for your specific kind of cancer. So it doesn't
Mm,
just come to you in one form sometimes, or other times, you might get three drugs, but it's all in the same IV. So it's going to be different for every patient. It's a very personalized disease. And
right.
that's what's so hard about treating the disease because, I left this out of my definition of cancer, but I think one of the most common misconceptions about cancer is that it's just one disease. Like cancer is actually a
Mm,
family of thousands of diseases.
right.
And I once or twice gotten into conversations with people about at bars about whether or not there's a cure for cancer that the government has under lock and key. And I
Right.
just think that's a silly thing to say because that's almost saying like, well, the government can also cure every kind of neurodegenerative disorder with one treatment. You know what I mean?
Mm,
Like take care of Alzheimer's and Parkinson's separately because they're different diseases. Like breast cancer and brain cancer are not the same disease. But then even if you look
right.
at breast cancer in and of itself, one woman's breast cancer is different from another woman's breast
Right.
cancer. And then going in further, you can have a tumor that has different mutations throughout
Mm,
the tumor. So it's a heterogeneous tumor that has different mutations and different characteristics. And half of the tumor might respond well to one drug, while another half of the tumor might be resistant to the same drug. So overall, it's just a very complicated disease that needs a very personalized treatment.
mm. Right. Like two people having the same disease or even the same stage of cancer will
Mhm.
respond to treatments differently based on their existing, immune system and
Yes,
how they, yeah, how the treatment works on them. So yeah. So I think,
right.
yeah, it's
Mhm.
very sounds very, very like in eye care we have this term. So we have customization and then you have personalization, and this sounds more like a very personalized treatment. So
Mhm.
yeah, I think that's why I think it gets really tricky to decide what kind of treatment is the best option for that particular patient. And that also I think takes up a lot of time from the patient, especially if the cancer is progressing
Mhm.
rapidly,
Right.
unless we
Unfortunately,
make.
yeah, because unfortunately, sometimes it might come down to a little bit of trial and error to see what works
Mm,
best for them. And you're right,
right.
when time is of an essence, like that's a risky thing to have to do. But that's why we want to do more research so we can make more specific chemotherapies and also understand how cells are going to respond to which chemotherapy.
Mm, interesting. Um, right. So before we start discussing, the big words actually, can you tell us a little bit about your research, what you've done, and what you're currently doing?
Sure. So let's start from just the methods I use. So I am in a structural
Mm,
biology lab, which
okay.
means that I study the way proteins are shaped to understand their function.
Mm-hmm.
So I actually am right now doing everything in vitro. So I don't even work with cancer cells. I work with proteins. So I grow and I express proteins that are implicated in certain cancer processes. And I study them using various methods to understand
Mm-hmm.
how they interact with other proteins or how they interact with drugs or how they can interact with DNA or so on and so forth. So when you look at kind of science, I almost feel like in vitro science is sometimes, I wouldn't say the first step because a lot of our research is based on cellular science, but I'm starting at the smallest stage. So I'm just looking at the itty bitty proteins that are inside of cells that make cancer cells act the way they do.
Mm-hmm.
So my lab and me personally, I study a process called translesion synthesis, which
Mm-hmm.
is the ability of our cells to copy our DNA, even if there is damage within the DNA. So usually if our DNA gets damaged, we have a group of proteins that are going to come in and fix the DNA. So that way we can replicate our protein and our DNA can continue doing its job. So coding for proteins and so on and so forth. But sometimes the DNA damage is too often in a cell and a little bit too overwhelming for DNA damage response proteins. So we have this kind of auxiliary mechanism where we can just copy over the damaged DNA. So then the cell can just deal with it later. And what it allows our cells to do is to replicate more and grow, even if there is stress during this time, rather than just having to kill the cell.
Mm-hmm.
So we have this in our bodies. It's something necessary for us to live and function. But unfortunately, in some cancer cells, translesion synthesis is upregulated. And when it's upregulated, it allows cancer cells to survive during chemotherapy. So what we want to do is create inhibitors for translesion synthesis. So that way we can sensitize cells to existing chemotherapy like cisplatin. So pretty much we're blocking proteins that can cause chemoresistance to really common chemotherapies. And that will allow cancer cells to die and for the chemotherapy to do a better job.
Okay. So, has this already been, is this already been practiced, or do we see this applied to some of the treatments that we have right now?
So not yet. So there are not any, there are no drugs that have progressed yet to the human stage. So it's very much a new idea.
Okay.
So we know that there are humans who have resistance to genotoxic chemotherapy. And if you were to genotype or look at the protein expression of their cancer cells, these translesion synthesis proteins are either upregulated or mutated in one way or another. So that's kind of evidence that something weird is going on. There's this really strong correlation between these proteins acting strangely and cancer chemotherapy resistance. So I would say that the stages that this field is in right now, because although I do study developing inhibitors for this pathway, there are other labs who also study inhibitors for this pathway. It's pretty, I would say we're still in the nascent stages of the drug design. And so there are 4 or 5 labs that I know of who have designed inhibitors for a protein within this pathway. And we have been able to show that it helps in vitro, so in dishes, cancer cells become more sensitized to chemotherapy. And there was one lab who was also able to show it in a mouse model, that if they were able to treat the mouse with a chemotherapy and an inhibitor, that the mice tumors would die quicker. Right? So it's very much the nascent stages where it's going to be a long time before any of these inhibitors are going to be introduced into a human model. You have to very thoroughly show that you have a good drug, both in vitro and in a mouse, before you can progress to doing that in humans.
On humans, yeah, right. I'm guessing the application for clinical trials would undergo much more scrutiny in your field, in comparison to the others that we have.
And chemotherapies is the drug that has the highest fail rate in clinical trials, right? And it's when you think about it, like chemotherapy is almost like a controlled poison, if you don't, if it's not specific enough, or if you're not using the right dosage, it is going to harm the human body. So it is a very, it's a hard drug to really push through clinical trials.
Right. And why is that? Why do you think it's poison? Is it because of some of the ingredients, or is it just the procedure we just spoke about? Like it could harm your body.
It's just that challenge of specificity. Like it's so hard to design a drug that's only going to affect a cancer cell and not affect anything else. And it's really hard and to jump from a mouse to a human is a
Right.
pretty big jump, but that's right now, that's the pipeline of how we design drugs. And, there are a few really high end research institutions who might be able to use something else between mice and humans. Like I know that
Hmm.
monkeys can are sometimes used, but that's more for vaccinations and what have you. Uh, what works well in a mouse is not guaranteed to work well in a human.
Right.
And so
Exactly.
it's a
Yeah.
yeah, yes, that's a huge difference. But that's the progression that we use. So, you have to have a very thorough study showing that a chemotherapy, or even another drug works well in a mouse before you move on to a human. And
Hmm.
most chemotherapies will fail in either the first or second stages,
Mm.
and it will be
Uh,
because of toxicity.
right. Um, and so, like, again, going back to chemotherapy and radiation, is chemo the last, is it the last step of treatment? Like, I know for a fact that the early stages of cancer, they prefer to use radiation instead of chemo. So
Mm.
is it usually the drug of choice? Is radiation the first preference? And then, like, if someone's diagnosed in the later stages, I don't think they expose them to radiation. They directly start with chemo. Is that the
Yeah.
preferred norm?
So radiation is less, what's the word I'm looking for? I guess it's less harmful and has less side effects in comparison to chemo. So, like, for
Hmm.
example, like my grandmother who's in her 90s, when she had to get some cancer treatment, they didn't want to give her chemo because they didn't know
Right,
if she could handle it. So they only gave her radiation.
right,
But
right.
unfortunately, the radiation only goes so far. So, like as we said
Mm,
before, it's very personalized. So from the beginning, there are plenty of people who will get chemo from the get-go alongside radiation or without radiation. And it really is going to come
mm,
down to the person, the type of cancer, and the severity of cancer as well. But
mm,
a benign tumor that seems to be pretty well contained, you might be able to get away with only using radiation. But
mhm,
plenty of scenarios from the beginning, you will also get chemo. It's not always that it's just radiation at the first step.
mm. There was also another question, this is, I think, going back a little to the environment. Now, is it possible for a person who's not genetically disposed to any kind of, say, to one particular type of cancer, and then you put them in an environment, uh, for instance, people in the West, so are more prone to skin cancer because of, you know, the lack of melanin and things like one of the reasons being lack of
Yeah.
less amount of melanin. So when you pick someone from South Asia or Southeast Asia, and then when you put them in these conditions, they have good amount of melanin in their body. So are they prone to skin cancer now because the environment has changed, or is that does that still act as a protective factor?
Yeah. So I get it's similar to what I said before, that it's really hard to just like tease out environment and genetics.
Mm-hmm.
So I think anybody can be environmentally impacted by UV light. But you are right that certain people are going to be more sensitive to UV light than others. So yes,
Mm-hmm.
the person that if you take from one environment and put them in a new environment where they're more exposed to a carcinogen, they'll have an
Mm-hmm.
increased exposure to cancer. I mean, an increased chance of getting cancer, but it doesn't necessarily mean they're going to get it. Like everything is going to come
Mm.
down to probabilities in that scenario.
Okay.
Yeah.
Okay.
And,
Got it.
in the end, I think it's something about like one in every three people. Is it one in three? I think it is one in three people will get some sort of cancer in their lifetime. And the majority of the
Oh,
people will
wow.
get it towards the tail end of their lifetime, but that's a pretty big number.
1 in 3 is a big
And
number. Yeah.
yeah, one in three is a big number. And another thing that actually happens in our bodies that I think is actually quite amazing. You and I have probably already had cancer to one extent or another, but our immune system recognized it and took care of it.
Mm.
And that actually
Mm.
happens quite a bit where you have these precancerous cells forming, but our immune system sees there's a cell going haywire and clears it out.
Mm.
And that's another thing that is a barrier for tumors to form, is that they have to form without our body noticing that it's
Mm.
growing. So it's another I don't know, kind of evil trick that cancer cells learn how to do is they learn to evade our immune system. So,
Right.
we're all, and in one way or another, we're almost predisposed to getting cancer, but then we have other mechanisms in our body that also help us make sure we don't get cancer.
Right.
Right.
It's only
And
when
those,
it goes
yeah,
haywire and out of control that is when you see the manifestation of
right.
the symptoms and things. Yeah. Mm.
Yeah,
Yeah, we were talking about translation synthesis and your research. Can you give us more details about that before we move on
sure.
to the other
Yeah.
type of chemotherapy? Yeah.
So I actually like translesion synthesis as an area to study. I think it's a pretty interesting pathway because, so as I
Hmm.
explained before, and I'll just reiterate this. So translesion synthesis lets us copy DNA that has been damaged, and it
Damage.
allows us to replicate our cells quickly, even in a stressful environment, instead of all those
Hmm.
cells dying. And that comes in handy a lot, like especially during development and what have you. There are certain times where it is very necessary for our cells to replicate, even if it's under stress. And that's why we have the cellular mechanism. But then another thing that is kind of a downside to translesion synthesis, other than it being hyperactivated in cancer cells, is that when we use this pathway, your cells are more likely to mutate because it's not
Hmm.
as good as the plan A and plan B, which is to uh, repair your DNA and then replicate it normally without it being damaged.
Hmm.
Plan C, which is translesion synthesis, is kind of almost like a band aid. It's like, let's copy the DNA really quick and then we'll take care of it later. And because of that, you're more likely to get a mutation. So if you're using translesion synthesis too much in these cancer cells, it can also cause cancer to accumulate more mutations. So not only does translesion synthesis let cancer cells evade chemotherapy treatment, it can also make your cancer cells more malignant, right?
Mm.
Which is
Yeah.
scary. Like you already
Right.
have these cells that are going haywire and not doing
Right.
their job. The last thing those cancer cells need are more mutations, which can make them do even more
Yeah.
crazier things like spread
Right.
or soak up more energy or even further become more resistant to other types of cancer chemotherapies. So that's the second reason why we're looking into disrupting this pathway,
Hmm.
because you want to stabilize these cells. You don't want them to get more malignant as they go on throughout the process.
Mm.
Mhm.
Interesting. Yeah, sometimes, medicine shouldn't be like, it shouldn't be causing more discomfort than like, even if it doesn't provide any kind of solace, it shouldn't be causing any more discomfort. Yeah.
Mhm.
Um, can you tell us a little bit about genotoxic chemotherapy?
Sure. So
Yeah.
cisplatin, which I was talking about before, is a version of genotoxic chemotherapy. So that's just
Okay.
agents that will attach to DNA in order to damage the DNA so the cancer cell cannot replicate and the cancer cell has to die. And
Mm,
radiation is also a version of genotoxic therapy. It's not chemotherapy, but it's genotoxic
mm-hmm,
therapy because it breaks DNA in half.
mm,
And so cisplatin and radiation are the two that we've been talking about the most, but there are plenty of other drugs that do a similar thing where they attach to DNA to make the DNA break. So that way cancer cells have to die. And overall, that's the most common version of chemotherapy that's in use is genotoxic chemotherapy. And there
mm-hmm,
are other versions of chemotherapy which are common, like hormonal chemotherapy or certain inhibitors that bind to specific proteins, as I discussed before. But the interesting thing about these chemotherapies, genotoxic chemotherapies, is that a lot of them have been in use since like the 50s and 70s, and they're still in use today. So we
mm,
have a lot of advancements that have been happening in the cancer field, but like I said, cancer is such a broad family of diseases that unfortunately there are plenty of subtypes of chemotherapies that we still really don't have targeted treatments for, and we just have to use these blanketed genotoxic chemotherapies.
right.
Mhm.
Um, yeah, that reminds me, how has the treatment, changed over years? Like,
Mhm.
I would say probably in the last 20 years, like maybe before 20 years, I don't think we've heard a lot of people dying of cancer or like suffering from cancer. But at least in the last 2 decades, we know at least one family member or a friend who've either had cancer, recovered from cancer, or like died because of cancer. So how has the treatment changed over years? And do you also, because I also see this, that the more you research about the study, you get to see, or more, the more treatment options that are being available, we also are seeing advanced cases of cancer, which
Mhm.
maybe because they weren't detected before they were there, or it's just because now we have the technology, we're able to detect them, or is it changing with the environment that we discussed? So what is it? I know
Right.
it's a bit chicken and egg,
Yeah.
but yeah, I think you'd be the perfect person to answer this.
Yeah. So I think it's hard to say, like you said, it's really hard to say if there are new cancers being formed because
Hmm.
two things will go into that. One, like you said, we have better technology now. So now we're
Hmm.
probably able to tease out a little bit different subtypes of cancer. Whereas before, if someone had just an organ cancer, we would say that person has breast cancer, not this person has triple negative breast cancer caused by a BRCA2 mutation. And so our
Hmm.
like it's hard to decide
Right.
if we have new cancers forming or if it's really just more that technology is better and we're learning more about the disease and we're able to genotype more and what have you. And then the second thing that's also happening is that lifespan is increasing. Because lifespan is increasing, the likelihood of people getting cancer are more likely because the older you get, the more likely you are to
Mm.
get cancer. So we're going to see more of it. Similar to how, like, because people aren't dying of, as many infectious diseases anymore as they used to hundreds of years ago, you know,
Mhm.
Alzheimer's is also increasing because people are just living long enough to experience it. So I wouldn't say that really there are new cancers forming. We just understand that it's more complicated disease than what
Mhm.
was initially concepted. But you're right in saying that, survival and the prognosis of cancer is improving. So I think about in the 50s, breast cancer had a 50% survival rate. Breast
Mm.
cancer now has about a 92% survival rate. That's a
Wow.
huge difference. So
Mm.
it went from a coin flip to you're probably going to be okay.
Yeah.
And for the
Yeah.
breast
Yeah.
cancer patients who do die, it's because they had a very aggressive version of the disease, or maybe they had a late, they only discovered that they had breast cancer later when it was already, in stage 3 and it was harder to take care of. So that's a huge difference. And I think that the biggest difference between cancer as we understood it before and cancer as we understood it and as we understand it now really comes down to personalized medicine and understanding the differences between different tumors and the different treatments that should be given to a specific tumor.
Mm.
So, it's not that everybody just gets surgery and the same chemotherapy anymore. If you have the cancer, likely you're going to be biopsied. Your cancer likely will get genotyped. And in cases of breast cancer, for example, you'll see what hormone receptors are being expressed and so on and so forth. So
Mhm.
that's the biggest change in the last like couple decades really comes down to personalized medicine as well as increased advances in genetic sequencing. Those are the two
Mm.
biggest differences that have made cancer be a lot more of a treatable disease. And especially in the case of breast cancer, as we discussed, that 50% survival rate being flipped to 92% survival rate in
Yeah.
50 years, that's the difference. That makes it to be a lot less scary and much more of a disease that you think that you can fight and that you think you can survive.
Mm. Yeah. And is it the case with all the cancers? Like you see that when you take the last 50 years, there's been rapid changes. I think because of the availability of computers or affordability of treatments and um, basically more opportunities. You can see that there's been a rapid change in life, or in career opportunities and research, of course. Like take COVID medicine for example. Like we have a vaccine in less than a year, which I don't think we would have dreamt of for other vaccines. So has that happened in the field of cancer too? Like in the last 50 years, do you see that in the last 20 years you've made rapid progress in cancer research?
Hmm. Yeah, so I
Mm.
think that, and I don't have a statistics to confirm this, but there's a lot of funding that goes into cancer research on numerous levels. So you have cancer research occurring from humans all the way down to the little itty bitty proteins that I do. And
Mm.
it is quite fascinating to see the advancements that we have made in cancer research, especially in comparison to some other diseases like Alzheimer's or Parkinson's, which haven't really progressed as much as we had hoped in the last few years. Um, and especially because when you think about how slow biomedical research is and how careful we have to be to ensure that these chemotherapies work, like we have absolutely made huge advancements that have, changed the way that cancer is treated.
Mm. That's the yeah. And you know, we were talking about translation synthesis and like chemotherapy, genotoxic chemotherapy, and the research that's ongoing. So how does this translate into treatment and medications? Like, you know, how does this, how does the pharmaceutical companies take this up, and like, how
Mhm.
does this translate into treatments, basically, in simple
Right.
words?
So the end all goal of this translesion synthesis research is to make an adjuvant chemotherapy. So adjuvant means it's a drug that's given alongside another drug. So
Hmm.
blocking translesion synthesis would allow drugs like cisplatin to do better. So if you had a cancer cell and you were going to be treated with genotoxic chemotherapy, the idea is that you would be given cisplatin, but also a translesion synthesis inhibitor. And the translesion synthesis inhibitor would make cisplatin better at its job. So maybe you would actually have to use less cisplatin too, which is good because that could reduce toxicity. It
Mm.
would decrease the chances that your cancer would become resistant to your chemotherapy. And it could
Mm.
also prevent that cancer from accumulating mutations, which could make it metastasize and become more
Mm.
malignant. So that's the end goal. So I'm not sure if any pharmaceutical companies have, often pharmaceutical companies will work with academics to improve the design of their inhibitors. I'm not sure if that
Mm.
has happened to any of the academic institutions studying translation synthesis yet. I'm unaware if any pharmaceutical companies are looking into this now, but I think in the future that you will start to see collaborations with the laboratories who are studying translesion synthesis and pharmaceutical companies. So right now where the inhibitors are at, both in my lab, but also in other labs who are studying this, they still need to be optimized. So when you first make an inhibitor for a protein, you want to make sure it binds to that protein and that it has a good binding affinity so that it's likely the protein and the drug will find each other in a cell and interact so that way the protein doesn't work anymore. But it takes a couple of rounds of optimization of that small molecule inhibitor until you have a drug that is likely to work inside of a multicellular organism.
Mm.
So when you have a drug, you have to make sure it's soluble, that cells will uptake it, that it's going to be stable, and that it's going to bind to your protein of interest, and that it's not going to bind to the other thousands of proteins in the cell, because you only
Mm.
want it to affect your protein of interest, not other ones. So that
Right.
is what scientists are currently working on optimizing. So we are
Mm.
optimizing the inhibitors and trying to make them as good as possible so they are very likely to work well in a multicellular organism. And
Mm.
that's where we're at right now. So
Okay.
it's going to be some time before we'll progress to that adjuvant chemotherapy, which is the final goal. But these stuff take a while, and there's a reason
Right.
why it does, because you have to make sure that you're not
Right.
going to hurt somebody.
Right.
Yeah.
On an average, if I may ask, so how long does it take? Like, I understand that, you know, you work at the cellular
Yeah.
level, so you're seeing the proteins and how that works. So the next step would be to probably, you know, apply that to or like inject that in a rat model and then see how that works. And then the next step is to probably do a human clinical trial
Mhm.
with whatever result we have, and then after that would probably come the pharmaceutical
Yeah.
translation. So right, like when you start the research and to see the end result? So what would be like a, I know it's not tied down, like it's not like a definite number, but how long does it take for that to happen?
So on average, from conception to clinical trial approval, so like we're
Hmm.
talking clinical trial 3, it takes about 30 years for a
Oh, wow.
chemotherapy to go, right?
Wow.
And it sounds like a long time, but there are so many things that can go wrong in the process from
Right.
conception until clinical trial approval. So,
Mm,
you know, so this drug is probably like this class of drugs that people are working on might be like 10 years in the making already, where people have already conceptualized it, studied it in cells, and come up with the idea of inhibitors. Uh, but and for the other thing too that you have to understand is that there have been multiple inhibitors that have done well in vitro, so in a test tube. So maybe there are like 30 that have been published, just to say that is a blanketed number.
hmm.
A lot of those are going to be discovered to be toxic or to not be able to be taken up by cells, or maybe it's not soluble enough and we couldn't optimize it to make it soluble enough to be good in a cell, I mean in a person. So
Mm,
plenty of those inhibitors that have been studied are not going to progress very far. So,
wow.
it's you need to come up with a bunch of different inhibitors targeting different proteins within the same system. And then they're going to slowly start weeding out because they didn't do their job in one way or another before we
Mm,
end up with we think is going to progress up through clinical trials. And then, as I said before, most chemotherapies will fail in phase 1 or 2 as well.
mm,
And then when they
right.
fail, you go back to the drawing board, you try to figure out why they failed, why did they become toxic? Can we change that so that way we could try again? So that's why it
Mm,
takes so long, because it's an iterative process at all the
right.
stages. You always have to go back and optimize and try again and try again and try again.
Mm,
Mm.
wow. 30 years is definitely a long
Yeah.
time. Yeah. Um,
Especially when you were saying that, we came up with a vaccine in a year.
yeah, exactly, right? Like, I know, like, we were able to come up with a vaccine in less than a year because there's already been research going on in the field of
Yeah.
adenoviruses and the coronavirus,
Right.
maybe at the lab level. So, which is why I think we were able to do a good job. And the amount of time, manpower, and
Mm.
funds spent on this particular project was, I think, pretty much everything took a back seat, and everyone had to concentrate on this. So, but yeah, like 30 years, I can't even imagine that.
Yeah.
Um, we were talking about resistance, and that kind of made me think about this. Uh, do these kind of treatments also behave similarly to how we have bacterial resistance? Like,
Mhm.
your body gets used to it gets resistant to some kind of a medication. So does that happen even in cancer and cancer treatments?
Yes. Yes, it's actually very similar to bacterial resistance. So you can almost look like a tumor as it being its own like ecosystem. So all these
Mm-hmm.
cells are, living and supposedly thriving, and then you introduce the chemotherapy, and it's going to kill a lot of the cells. But all you need is a few cells to divide and get a mutation that lets it survive in the site of the chemotherapy. So then
Right.
that cell and I mean, that's evolution, right? So you have a
Right.
cell that mutated and then it adapted to its environment. And unfortunately, that environment is the chemotherapy, which is supposed to cure the
Mm,
cancer. And now you
right.
have a new subset of cancer cells that are not responding to that chemotherapy.
Mm,
So that is exactly what happens, similar to bacterial resistance. So if you had a bacteria that was being treated with, I don't know, penicillin, and say there was a colony of thousands of bacteria, but there was one bacterial cell who mutated, got a mutation, and then that's going to colonize you instead.
mm,
So,
mm,
it's like survival of the fittest. If a cancer cell gets a mutation that allows it to survive, not only is that cell going to survive, it's going to start replicating, proliferating, and
right.
then taking over the
Mm,
cancer tumor, because is
right.
what's going to survive to the drug. And that is one of the reasons why you usually get a cocktail of drugs to try to avoid that, because it's
Mm,
easier for a cancer cell to mutate and resist one drug rather than two.
And does it also become autoimmune? Are there chances?
Yes, that's another thing it does too. And that's something that
Wow.
has to overcome earlier on in its stages, is to find ways to mutate so our immune system does not recognize it.
Hmm.
Mhm.
Wow, that is just when you think you have an answer, you have more questions. Right. I remember you telling me this last time we spoke about, when you talk to people about cancer research, and they're like, "Oh, okay, do you have an answer to this? Like, is there a cure to cancer?" How do you answer to that question? Is there really hope? And I mean, there's definitely hope, but is there a cure for cancer?
Okay. Is there now, or is there will be?
Is there now, and how are the chances?
Yeah.
Like, how is the future, like in the
Right.
future? Yeah.
Okay. So as I said before, there is currently no cure to cancer, and the government cannot cure cancer. So that is a conspiracy theory that cancer can be cured, but the government needs the working class to die off or something. And no, that's not the case, because as I said before, cancer is a super complicated disease,
Right.
and it's not just one disease that can be cured with one pill.
Mm,
And I think it's even difficult to say that in the future or in our lifetime we're going to see a cure to cancer. And what I like to describe is, to compare this to, is that there's only been two diseases in the history of mankind that have been eradicated from the planet. And one of them is a virus that actually infected animals, and the other one is smallpox, which was eradicated
smallpox.
due to
Mm,
vaccines, excuse me, power of
mm,
vaccines. We don't have smallpox because of vaccines.
yeah.
Um, we still don't have a cure for the common cold, for the
Yep.
flu,
Yeah. Yeah.
uh, all the neurodegenerative diseases, muscular dystrophy. I can go on and list all on and on and on.
Hmm.
If for some reason cancer gets this bad rap that it should have been cured so long ago, and scientists aren't doing their job, or there's conspiracy theories out there. And what I like to say to that is, we likely won't see a cure in our lifetime, but what we'll see is treatments, and we will see evolving treatments, and we will see better chances of survival. And again, I'll use
Mm,
breast cancer as an example. It used to be a flip coin if you would survive breast cancer, and now it's 9 out of 10 women who get breast
right.
cancer will survive it. And that
Mm,
is the trajectory we're going to move towards with all different kinds of cancers. So hopefully, the more difficult cancers like ovarian and pancreatic and brain will start seeing more personalized medicine and increased
mm,
numbers. So it's unfortunate that it's not going to be an end-all cure where you just have a pill and wake up the next morning and you're better. For some time to come, cancer will be a process that you have to work through in order for your treatments to work, but it is a survivable disease. It's not an end all death sentence like it used to be.
mm. And having said that, what is it? What does it mean to be cancer free? Like, especially after going through treatments. So, what
Mhm.
is defined cancer free right now?
I suppose cancer free means that you're eradicated of any detectable cancer cells.
Mm,
And it's a difficult thing to definitively say that you're cancer free and you will be forever. Because if you had
mm,
cancer, you were probably predisposed to have it for some reason or another, and it might reoccur. But
mm.
to be cancer free is that you don't have any detectable cancer cells
At present.
at present. Yeah.
Mm, okay.
But I would refrain from going to say it's an end all, like, it's completely over. You never know what's going to happen, later
Mm,
down the road.
right. Right. And again, like coming back to a bit about the different types of cancers. So why is that some of them are rapidly progressive and some of them, like the cells again?
Mhm.
I understand that ovarian cancer is like it's rapidly progressive, whereas maybe breast cancer,
Mhm.
and like prostate cancer in men, that's less progressive, in
Mhm.
comparison to other things. Again, does it have to do with the protein function, or the rate at which the cells are being produced? Is that what it is
Yeah.
about? Mm,
So, a couple things there. One is, it is always going to come down to the nature of the mutation which caused the cancer to progress.
Mm.
And some cells are more predisposed to having a certain gene being mutated to cause that cancer.
Mm.
And as luck would have it, that gene that causes cancer in that organ will cause very rapid progression of mutations. The other thing too is that unfortunately some of the types of cancers which are more severe are internal organs that cannot tolerate as much growth as an external organ, for example. So if you
Mm.
have, for example, pancreatic, you don't need a very large tumor for you to start feeling, for your body to start having side effects for that cancer,
Mhm.
because it's the nature of the organ that it's small, and if you have a tumor growing on it, it can press against it and make it the organ act all haywired. And then another issue too is detection. So
Mm.
we keep using breast cancer as an example, but it's one of the cancers that we do know the most about. Breast cancer can be detected. Colon cancer, even, we have time set up where it can be detected and so on and so forth. But stuff like pancreatic and brain cancer is often not detected early enough for
Mm.
us to do much about it because it's not as easily screened. So that also
Right.
comes down to prevention. But when you ask why do some cells progress and proliferate more than others, it is going to come down to the nature of the genetic mutation.
Mm.
Mhm.
Interesting. Um, yeah, so I now want to move on to, the other aspect, which is the science communication. But before that, I want to know if there's something that you want to add to our viewers, just to tell them more about cancer research and
Yeah.
anything about what we spoke. Yeah.
No, I think that we covered most of everything. Um,
Okay.
yeah, I think that we're all set there.
Okay, that's good. So now moving on to something which all of us are interested in, or because we're both kind of are interested in this kind of practice. So I usually ask my guests this question, especially those who practice science communication, uh, because there's no hard and fast definition for this. So what is science communication according to you? Like, what is your definition of science communication?
I have a much more broad definition than some science communicators. I think it's any time that a scientist at any level speaks about or communicates science to other people.
Mm.
So I believe that scientist to scientist communication is a form of SciComm, whereas other
Mm.
people might say that science communication is only communicating science to a general audience. But I think
Mm.
that, communication happens on all levels to all
Right.
different audiences. So science communication can be scientist to scientist.
Right.
It could be scientist to scientist in different fields, like you and I are doing right now, just talking to each
Mm-hmm.
other. But it can also be,
Right.
scientists to the general public, scientists to children, scientists to high school students. It's any times that a
Mm.
scientist sits down and makes an effort for their research to be understood by a specific audience.
Mm. Right.
Mhm.
And how did you get into this? Like, how did you choose to do this, and how did you come to know of this?
So I think that during my master's program, I really realized that I enjoyed writing and talking about science more so than doing it.
Mm.
Some people love working at the bench and doing data analysis and the whole journey of research. And it's not my cup of tea. I'll put it
Okay.
nicely. But
Uh-huh.
I like having the results in front of me and then talking about those results and how those results, one, should be communicated to tell other people, but then also to discuss the implications of that research. That's what I like
Right.
about science.
Mm.
So when I went off and to go start studying my doctorate, I knew from the beginning that I wanted to do something either working as a medical writer or in science policy or as a medical science liaison, which are all different jobs that utilize science communication.
Mm-hmm.
And I was given advice by someone who worked in science policy that if I wanted to work in that field, I had to start, kind of drumming up a CV and showing examples that I like writing. And it was actually recommended to me that I could start blogging. That is
Mm.
something that I could do to, one, hone my skills as a science for me communicator, and two, to also to make myself marketable for after my
Right.
PhD.
Mm-hmm.
So my first inclination to start doing science communication actually really came from a selfish standpoint where I'm like, okay, well, I need a job. I need to start working on myself and doing my CV.
Right.
And that's totally was my inspiration for starting my blog. But then when it came down to my blog, the question was, what am I going to write about? What is my brand? How am I going to build this blog? And two things I thought of right away was one, I don't know if I'm going to have time to do this.
Yeah.
And two, yeah, I don't know if I'm going to have time to do this. And two, I wish that there was already like a blog just set up that I can contribute to rather than me having to write every week or every other week, which would take up a ton of time. So I started looking online to see if there were blogs that I could just periodically write for, and I couldn't find one. Actually, I did find a couple, but one of them I didn't like the branding for, and then the other one actually I had to pay in order to write for them.
Oh, okay.
Which I was like, that's weird.
Yeah.
So that's why I started my blog and I made it collaborative, because the way I thought of it was that there must be other scientists out there who, like me, don't want to write for blog posts a month or however often you need to in order to keep a successful blog. And two, it actually ended up being really good because then the collaborative blog makes my blog much more diverse. Like you wrote for me and you're an optometrist. And, so because of you, I have a piece of work on my blog about vision science. I would have never written a post about vision science. I would have just wrote about cancer over and over again until people were bored of it. So it ended up growing into a platform where I get to work with other people instead of working with isolation, which I really like. So, you asked me, why did I start science communication and blogging? It totally came from wanting to have career development during my PhD for the specific career I wanted to. But the sidebar from that is I've learned a lot about the advantages of science communication, why it is important. And my blog started doing really well during the pandemic. And one of my first biggest posts that did very well was I wrote a post about hydroxychloroquine, right? So I think that, so that was a blast from last year. There was a month where chloroquine was constantly in the news and we don't hear about it anymore. And I wrote that piece, and that was one of my first pieces that had a lot of reach to a non-educated audience. So a lot of my blogs reach other scientists, but this reached a lot of people who I haven't spoken to in years and don't have a biology background, and they read the post. And that was amazing to be able to educate people about something that was going on in the news and a really big a really big issue in science that was impacting people. And it felt really cool to write science and in a accessible manner where people were reaching out to me and they were like, thank you so much for this. Like, I didn't understand what chloroquine was, but everybody was talking about it. So that was a really great experience for me to see that science communication is important and that we need to talk about it. And science isn't something that's just done in a lab. It's something that impacts other people.
Right. And I think now more than ever, you need more people to talk about science and the importance of science. I've seen a lot of change in people. I mean, we do have sort of people who still don't believe in vaccines and science and things, but we've also seen a lot of people trusting in science and understanding the importance of talking about science, thanks to the pandemic.
Right.
Yeah, and you write about, those articles. I still hear from people about the article which I wrote like maybe 8 or 9 months ago. I still get people emails from someone who read the post on your blog, and they'll be like, yeah, I read your post and I had this question and I was going through this and I saw floaters and I saw this, so I thought I'll talk to you about it. The other day, one of the students from Bangladesh, he sent me a really long email. He was trying to get in touch with me, but he texted me on Facebook. Then I on Twitter, and then I think on your blog post. And then I sent him my email address, and he sent me a really long email about his observations. He's probably like 18 or 19, and he's done quite a bit of research, and he wanted to discuss about it. I was like, yeah, let me make time for this and get back to you. But it was really nice that, this 19-year-old reached out with his observations, and I was really impressed. And really, thank you for giving me a platform to write,
Yeah.
on your blog. Um, yeah, so can you tell us a little bit about your blog, The Bolded Scientist? And I think you've had about 50 articles. Like I was just
Yeah.
skimming through that, and they're pretty diverse. Like, yeah, you have scientists writing about not just the science, like the science about the proteins. That you've written a lot of blogs about your research, and then there's a lot of active COVID research articles that happened in the
Mhm.
last 1 year. And I've also, I was able to get in touch with some of them through your contacts. Um, and I also found a lot of plant research, which it's not the first instinct for me, at least, to actually go back and look up plant research, but there were a few things about that too. It's pretty interesting. A lot of blood, like related to blood and things like that, and also maybe a little bit about science fiction and science culture, which was pretty interesting too. And I also really liked some of the articles about being a graduate student and how life of a graduate student, and like the burnout theories and things like that, which I think should also be communicated in addition to all the science that we talk
Mhm.
about. So can you tell us a little bit about your blog?
Yeah. So pretty much the way my blog works is there's a blog once a week. Right now we're in summer hours, so we're only doing every other week because I need a life.
Yeah, right.
But yeah, so the way it works is that any scientist who wants to contribute to it just has to email me with a brief pitch. So usually someone will email me and say like, "Hey, I came across your blog. I'd love to add to it. This is what I want to talk about." And I would say that the most common pitches I get from people is someone saying they're going to write a SciComm piece about their own research. So pretty much they're going to take their super complicated research and they're going to boil it down to a very digestible, readable, small blog post and explain it. That's probably the most
Mm,
common pitch I get is like, I want to tell people about my research in an accessible manner. And so I get that pitch a lot, and then I'll get a pitch about life as a scientist, which I think is important
mm,
because there are people doing this work. This work just doesn't exist as
right,
an entity in its own. And there are different
right.
kinds of scientists, and there are different paths to becoming a scientist. So I actually like one of my favorite posts was by some a writer named Jake, who wrote about how he became a scientist and why he is a scientist. And he explained that, for him, it wasn't about passion. It wasn't about this diehard need to be a scientist. It was more of, I'm a student in Malaysia and I need to find a way to be educated on a budget. And I also would like to travel. Like, how do I do this? And it ended up being that being a graduate student was his way to getting an education in Australia and being able to fund
Mm,
life abroad while getting an education. And then he explains that, science was a means to an end, but then he learned to love science in the process. And I love that blog post because I think that so many people sell this narrative that all these scientists are, die hard, this is their vocation, they love it, it's their life.
yeah,
But no, there are plenty of scientists like me who are like, no, this is just my 9 to 5.
yeah,
Like
yeah.
I
That's
yeah,
true.
I enjoy the work, but I don't, for me, it was never a calling. And I don't think that you should feel out of place if you are a
Mm,
scientist, you just work a 9 to 5 and it's not your, life's passion. I also think that's an okay thing to do too. So yeah, we get posts about being a scientist, posts about just your general research, or the next common thing is people will like to write to fight misinformation, which is something that absolutely
mm,
needs to be done right now. Because probably for every article that gets posted about science, there's probably 10 articles that get posted that are about, fake science and conspiracy theories and so on and so forth.
right,
And there is a healthy amount of research out there that shows that misinformation spreads much quicker on the internet than facts,
right. Mm,
right? Because let's be honest, like a conspiracy theory is more interesting than the truth. And
yes, yeah.
right. And so it's really important that I think scientists try every now and then to get out there and put out information and facts. And I have seen it where I've written an article or someone else has written an article about COVID, about vaccines, or about cancer, and people do read it and they're like, oh, I didn't understand that before. No one ever described it to me like that. So I think it's important that scientists do make an effort to talk about science in an accessible manner where it's not tedious to listen to, it's not boring to listen to, and it's easy to understand.
Mm. Um, can you tell us a little bit about women in science? I think you've probably written about that or you spoke about that in one of your interviews, I guess, if I'm not wrong. Yeah, can you tell us about how you like it could be a personal experience or it could be like an anecdote someone must have shared with you, but what is your opinion and how do you see it?
My opinion about what?
Women in science?
Oh, women in science. Oh, yes. So yeah, I've had a few articles about women in science, and I think right now you are seeing visibly more women and women in science. So like I think my department has quite a few women. Would you say that yours does too, where you work? Um,
Yeah, I think
would you
we
say
have
that
a
you have
good
a lot of
mix.
women in your
No, it's a good mix. It's a
yeah, I
good
think
mix.
that where I work, yeah, I think where I work right now, it's a decent mix, but then there are other departments that are predominantly men.
Mm,
So I'm fortunate where I work in a department where there's plenty of women, but like I have a friend in the immunology department, and I think there's one female PI out of like 20. And
mm,
there are also women are more likely to work in the biology fields, but they're less likely to be in the chemistry and the physics fields. So there is definitely an issue of
mm,
women in science. And there's been quite a few articles in my blog posts about that. But I think that one of the things that really affects women in science is, having to deal with motherhood in academia, which is really hard. And there are a couple of posts on my site about that, where there could be some institutional policies which can help women stay in science longer. And there was a PNAS article that actually said that 40% of new mothers will leave science either part-time or altogether. And that's a lot. That is a huge number.
huge. Yeah.
And it's not an easy fix, but I feel like that is one of the biggest challenges that women face, is when you have to decide between having kids and investing time into parenthood or having this viable, thriving career. And I would like to see it at one point where you don't have to make that choice.
Mm,
We had a women in science dinner recently for my department, and it was all the women talking about that. And I led the discussion questions, and that was the question I came up was about balancing motherhood and academic science. And one of the professors actually said, if you look at my CV, you can tell when I had my children.
yeah.
There's a gap
Gaps.
in publications. And unfortunately, especially since now that the trajectory to becoming a tenured professor is increasing with time. So like it used to be that, young professors might be able to get tenured in their 30s or early 40s. And now it's more mid 40s, late 50s that people are going to, mid 40s to early 50s that people are getting tenured. You need those formative years to publish, but you also need those formative years to reproduce.
Yeah.
So that is like that's the biggest issue for me that I would like to see changed, and I would like to see advocacy for, is to
Yeah.
have an environment that's more inclusive to new mothers and to young female scientists.
Hmm. Do you see a change in policies? Like have there been changes in policies, especially in this regard? Like the advocacy that you were talking about, yeah.
Yeah, I think that some universities have changes in policies where it's like allowing longer maternity leave and also having more flexible schedules for new mothers. What
Hmm.
I would like to see more of is actually to also include paternity leave with a P, because I think that both
Yep. Yep.
females, if you make it so that way men also take time off and invest in
Right.
parenthood more, then it can even the playing field for women, because then it's less of a
Right.
woman problem and more of a both sides of the spectrum problem.
Mm. Yeah.
But I
Interesting.
think that in the end, for academic scientists, at least, it's going to come down to publications. And
Mm.
even if the university supports, women taking time off and so on and so forth, you still need to have a publication in the end. So, and
Yeah.
that's really when it comes down to a problem, is how do you let women thrive in their career when losing publications is detrimental to their career, but they have to do that in order to have children. It's not an easy question to answer,
Right.
and
Right.
then there's always a lot of people on Twitter do talk about, this publish or perish or
Mm.
attitude in academia. Like, should we only be weighing the importance of a scientist and how many publications they have? But then
Mm.
alternatively, how do you judge how successful somebody is if you don't do that?
Yeah.
It's
Yeah.
not an easy answer.
Right.
Yeah.
Interesting. Yeah. They a lot of things to think about, isn't it? Um, yeah, I think I'm totally out of questions. Do you have any last thoughts or like anything that you want to add before we conclude this?
I would just say if you're a science communicator out there and you're watching this, feel free to reach out to me and I will put you
Yep.
on my blog. I'd be more than happy to. One thing that's really nice about my blog too is that you're not doing it in a vacuum. So I think it
Yeah.
would be
Yeah.
hard to have a blog on your own where you just put it out there and you just
Yeah.
hope you get a couple likes on Twitter. The thing that's nice about
Right.
working with me is that, we workshop the blog beforehand, we talk about it. So overall, it can be a pretty engaging and learning experience. So yeah, reach out to me if you would like to be featured.
Yep, I will put your Twitter feed, your Twitter link, and I'll also link your blog post
Mhm.
to or the web page in the description so that people can get in touch with you, um, if they're interested. And briefly, this is one question I think I kind of skipped out. If you want to understand this like from a like I'm interested in science communication, like this podcast is part of science communication,
Mhm.
but if I want to be trained in it, like are there courses or like can we take something, can we study about this to, because I know that science communication is has not been added as a course in most of the universities. Like I've not studied about science communication, I just happened to know about it because of Twitter and social
Mhm.
media. And I did all the research and figured out what is of more importance. Like I'm interested in writing, which is why I work for you and for another blog. But then I also I'm curious about a lot of other things, and I thought this would be one way I can talk to a lot of people, get
Mhm.
my questions answered, and get probably the questions that people are thinking about, get them answered too. But where can we go if you want to like probably take up a short course about
Mhm.
some science communication?
Right. So I'm similar to you where I'm mostly self-taught for science communications, but I have come across a few people recently who have either taken courses in it or even a minored or majored in it, which I think is
Oh,
pretty
okay.
fascinating. And I forget what university it is, but I was speaking to someone recently who applied to a program to get her PhD in science communication. And I was like, I wish
Wow.
I knew that was a thing
That's
when I was
interesting.
applying. Yeah, when I was applying to my PhD, I wish I knew that that was an option because I didn't.
Mm,
So I think that you'll see the field of science communication grow to where you do see more courses and more education. That being said, I do know there are plenty of conferences that are offered or workshops in order for you to hone your science communication skills. And I know one of the conferences is, I think it's called ComSciCon,
mm-hmm.
and they do workshops and conferences across the United States, maybe across the world, but I'm not sure about that. I know it's across the United States. And I'm sure if you were interested in honing your skills, that there are other forms of conferences or even fellowships that have that available to you. But I would say if you want to learn more about it and you don't have time to take a course, or I know for some of the workshops you have to apply to, you might not be able to get into them right away. You
Mm-hmm.
know, do what you and I are doing right now and educate yourself. There are plenty of people
Mm-hmm.
on Twitter out there who not
Yeah.
only participate in SciCon, but will actually talk about writing skills and communication skills and so on and so forth. And
Mm-hmm.
there's a huge dialogue out there in Twitter about science communication. For me,
Mm-hmm.
that's where I learned most of my stuff and practice. And I think you
Yep.
would,
Yep.
wouldn't you agree to that? Like your writing and your communication has gotten a lot better just
Yeah.
from, doing these side gigs.
Right. Right.
Yeah.
Oh yeah, definitely. Yep. Yep. I mean, like if I look at my first video, there's definitely I'm not done enough, but I can definitely see a lot of change in the questions that I'm thinking about and
Uh-huh.
the ways I can articulate even more in probably to the general audience and also let the speaker get come because not all of the speakers that I've had were science communicators. So some of them are like hardcore scientists who are like very prone to using jargon. So I had to make them aware
Yeah.
that this is for both the audience is both general people and the general public and scientists. So they were like, oh yeah, that is, I've never done that, so this is going to be a very good experience. Um, yeah, I was also kind of interested in looking up courses online, but I wasn't sure which one is like, worth it. And I had no idea about this PhD in science communication. It's
Yeah.
pretty interesting.
Yeah, I wish I could remember the university. It was someone who actually had written for me previously who had said that she had applied
Mm,
to a program. And I was like, because I hadn't even heard of the fields of science communication until I was
right.
already doing my PhD.
Mm,
But which is fine because self-education is sometimes even better than going and
right.
being taught, because sometimes you learn more through trial and error and mistakes and like figuring stuff out.
Yeah.
Yeah. But
Mm,
I would like, it's funny, about when I came around to my year anniversary of my blog, I was actually going back and reading some of my older and earlier blog posts. I actually deleted 2 of them because I was like, oh my gosh, these are awful. I don't want
I
this
know.
being advertised as my anymore. And then a couple of
Right.
them I edited and I reworked in order to make them better.
Yeah.
So a year and a half of running a blog, of personally writing but also editing, has, definitely been a good experience for me, because my writing is definitely more skilled, and I'm learning to target better and write in a more accessible manner and a more enjoyable manner too. It doesn't always have to read like a textbook. So
Right.
it was hilarious when I went back. I was like, oh my gosh, I'm so embarrassed I wrote this and I posted this online and I deleted it.
Yeah.
So I think that
It's
definitely shows I've had growth.
Yeah, it's like going back to your Facebook post where you're like, seriously, did I post this?
Yes, especially like in middle school or high school, when
Yeah.
you get like a memory, like you said this 7 years ago, and it's like, well, I shouldn't have.
I know. That's true. That's
Yeah.
very true. So thank you very much. I think we had a very productive conversations, and I really hope our audience understand a bit about cancer. And I think you did a great job in demystifying the big words. And I honestly have learned quite a bit because, like I said, cancer was something that I know of, not much,
Yeah.
like at least at the levels of like the nuances and things like that. Yeah, I don't know, and they're kind of, I think, very important because like I said, everybody knows someone who's gone through this. So I think this would definitely be a good it they might find this very educational. So thank you very much for your time.
Mhm.
And yeah, for the general audience, we have many such interesting topics and speakers coming in the upcoming weeks. So please like and subscribe to Nothing is Rocket Science. And we would love to definitely hear from you. So you can feel free to comment in the comment section. And I will also be linking Kerry's details, like her Twitter handle and her blog post. So if you're interested to contribute or learn more, please feel free to go check Bolded Scientist's blog. They really have it's a cornucopia of science and science related stuff. So thank you once again, and nothing is rocket science. Stay curious.
All right. Thank you.